senhor tem Twitter? Então, tá certo.
Okay, Professor Antônio Costa-Filho has a very nice title in his talk, To Be or Not to Be, What's the Function? Exquisite Biophysics of the Golgi Reassembly Stack Structure. Floor is yours.
Thank you, Danilo, for the introduction, and I would have to start thanking you and Pablo for putting this together, bringing people together. And I have to say, well, as the other speakers of the day, that now this is a complete change of gears compared to anybody else during the day. And Lisa said that the equations were there to keep you calm. So I guess by the end of my talk, you'll be all nervous because there are no equations whatsoever there. Not yet, not a single one. So, and we will move down the side, the size scale to look at the behavior of molecules, biological molecules, particularly proteins. And the proteins we'll be interested in today is the proteins from this family called the Golgi reassembly and stacking proteins. So the acronym for this family is GRASP. So we can say what we want to grasp today. So this family of proteins, and I think by the end of the talk, we can find common ground between the physics that have been discussed here during this day, and the thing that you see there, I think that this is the main goal of this group of study of people, right? So last time I was here, we were convinced that it would be nice to bring physicists working in different aspects of the different systems so that they can build collaboration, find common ground to work together. So I think this is a very nice setup. Then we have physicists doing things from all the way from protein folding to what Lisa showed in the experiment. So, so this family of proteins, GRASP, they are mainly responsible. They were first identified as the structural factors of an organelle within the cell called the Golgi apparatus. So I believe that everybody has remembered, even from high school, what the Golgi apparatus is. So the Golgi apparatus is an organelle within our cells. So this family of proteins was believed to be responsible for the formation of this. You can see that there are some proteinaceous bridges there. So that's the stack. So the Golgi is an organelle within the cell where it's constituted by a pile of, a stack of cisterns. And these cisterns are brought together by this, it's kind of a glue there, right? So, and the Golgi apparatus is like the male, it's like the male broom of the cell. So proteins that are synthesized in the plasmic reticulum are directed to the Golgi apparatus. There they are chemically modified, and then they are dispatched to the final destination, either within the cell or outside the cell. So they are in the core of what is called the conversion protein secretion pathway, okay? So more recently, this family of protein was found to play an even more important role in the cell in the so-called unconventional protein secretion pathways, okay? So these pathways, they are turned on when the cell is submitted to stress conditions. So this is kind of a desperate measure that the cell takes to keep secreting things outside when it's needed and when the stress secretions are found. Okay, so this family of protein has been reported to be involved in two types of unconventional protein secretions, UPS. And one of them is kind of a schizophrenic type of behavior because it's the Golgi. Those are Golgi resident proteins that during stress situations, they go back from the Golgi to the ER, engage in some sort of carrier, nobody knows about that, and then they secrete things outside, outside the cell, bypassing the Golgi. So they are in the Golgi, and some, during stress times, they just leave the Golgi to keep secreting things outside. All right. So what we know about this family of proteins, the GRASP, so we know that they are constituted mainly by two domains, structural domains. Okay, so we're talking about a molecule. So that's in the nanometer scale, right? So the nanoscopic thing. So one domain is called the GRASP domain. This is a bit confusing. So the GRASP protein has a GRASP domain and also an SPR domain that's completely disordered there. Okay, so within this GRASP domain, there are two subdomains called the PDZ1 and PDZ2. So this protein is attached to the membrane of the Golgi organelle, and there when they this, the function, right? So we have information about the 3D structure of the protein. So that's obtained from X-ray crystallography, and we don't have the structure information for the whole protein, just for the order domain, the graph domain. Those are the two representatives of grafts in mammal. So we have two grafts, and they are called graft 55 and graft 65, and they are located in different parts of the Golgi apparatus, right? All other organisms have only one graft representative. So that makes a difference between us and bacteria, for example, right? So what we don't know, well, there's a lot of things we don't know. So we don't know how the graft proteins interact to form this glue within the right in between the cistern of the Golgi apparatus. So there are different models, the structural model nest, to propose how they bring together the cistern of the Golgi apparatus. And we don't know, well, we know very little about in terms of molecular details of how they play this role in UPS, right? So our group is, well, we are a molecular biophysicist group at the University of São Paulo in Ribeirão Preto, and we started this work by, well, it's very important to come to meetings like this one because you have a chance to talk to people from different fields and have a beer together, and then ideas come up, right? So this project came from a meeting that I went in Italy, and I was watching this talk by this guy called Márcio Rodrigues, who is now in the Fiocruz in Curitiba, in Brazil, and he's a cell biologist, and his group was interested in this fungus called the Cryptococcus neoformis. So they showed that the graft protein in this fungus was responsible for the secretion of the virulence factor of the fungus. So it's a sugar, you can see here is the fungus, this in there. And around the fungus, there is a layer of sugar. That's the virulence factor of the fungus. So they show that when they knock down the gene that expresses this protein, the fungus does not form this layer anymore. And we were talking. I said, "Oh, what do you know about this? The biophysical and biochemical properties of this protein?" And he was like, "Oh, we know nothing pretty much." So we started doing from scratch. He had the DNA to express and purify this protein in the laboratory. So all experiments you'll see, it's in vitro, all right? I will not go into the details of all the methods we use there. If you want to know details about them, we can discuss later on over coffee or over a beer. It doesn't matter. So we started by using a combination of physical chemical methods to characterize the structure of the protein. So that you can see results from magnetic resonance, circular dichroism, some prediction, bioinformatic prediction, some proteolysis assay that you can follow whether the protein is degraded or not by enzyme. And our final conclusion from this was that these graft proteins, they are not, let's say, well-behaved globular proteins. Okay, so they are in the different realm of proteins called intrinsically disordered proteins. This was a new result. So I don't know how many of you are familiar with this thing. So the way we think about proteins usually is that they are globular, round-like things that have a very well-defined 3D structure. So that's not the case all the time, right? So these intrinsically disordered proteins, they do have a 3D structure, but they can coexist in different 3D structures with different conformations, and that gives them a very nice set of tools to interact with other partners, right? So we proved that. And we went a little bit further and tried to find whether this property of being intrinsically disordered was also found in grafts from other organisms. So we started with the fungus, the Cryptococcus neoformis, and then we started producing, making in the lab the same protein from different organisms. So there you can see a phylogenetic analysis that we did using grafts from human grafts, the two we had, 55 and 65. Fungus one in the yeast. So I learned of, in the way that yeast is like the harmonic oscillator of cell biology. Okay? So the guys have everything you can imagine, all types of experiments you can do. The cell biologists do it with so they are like the harmonic oscillator. It's like putting the X square potential everywhere you find in it. So that's what the cell biologists they use. So we were like, well, does this have graphs? Yeah, they do. So that's the graphs from you. And we found out in this study that yes, all the graphs from different organisms are also intrinsically disordered. And surprisingly, using this phylogenetic analysis there, we found that one of our grafts,
looks more similar to the grass from the lower eukaryotes than the other. Okay, so grass 55, no, let's keep this in mind. So grass 55 looks different compared to 65 in the lower eukaryotes. It's kind of interesting, this or fun, if you want to, if you will, because one of the higher eukaryotes grass looks like the lower eukaryotes one, and the grass 55 is different. Okay, this information, and okay, we'll talk about this a little bit talk. So we dig it up a little bit more. So remember that I told you the graphs are formed by two domains, and one of the domains is formed by two subdomains. So we chop it off, right? So we can do that in the lab. So we cut off the disordered domain. In the ordered domain, we chop it off in the two subdomains to try to find where this intrinsically disordered region was. And we found out again using a combination of biophysical methods there, experimental methods, that's the NMR, that's circular dichroism. We found that the PDZ2 is the domain that behaves, let's say, properly, like a globular protein, in the PDZ1 does not. That's an interesting result, because if you look at the structure, the 3D structure you can find in the PGB of the two subdomains, PDZ1 and PDZ2, they look the same. But their dynamics is not, and the dynamics is determinant for the molecular interaction. So we found a biophysical explanation why there are so many binding partners reported for PDZ1, and just one binding partner reported for PDZ2, because PDZ1 is intrinsically disordered, and intrinsic disorder has everything to do with interaction. So intrinsic disorder makes the protein very promiscuous. Okay, so we could explain some results that were in the literature. Well, in the pursuing this biophysical characterization of grasps in other organisms, we start to look at the grasp from the east. So GIH1, right, you see that is the grasp from the east, right? And we are trying to study how this the protein structure changes when we change, for example, temperature, or you add a denaturing agent, or when we change the dielectric constant of the medium, trying to mimic the presence of a membrane. And we found out, this is the secret mechanism there, that when you push the protein to denaturation, to unfold the structure, instead of changing to an unfolded structure, the protein was changing to a structure that was rich in beta sheet-like structure, right? I guess that most of you don't actually know what that means, but beta sheet rich structures are found in proteins involved in neurodegenerative diseases, right? Which forms what is called the amyloid fibers. Of course, the yeast does not suffer of neurodegenerative disease. So we were like, well, so what does this? What the heck? I mean, what's going on there? Like, what does this beta sheet rich structure appear when you try to unfold the protein, right? So well, at the time, I told Natalia, who was a PhD student in the group at that time, said, well, look, let's see if they really form these amyloid-like structures. And she showed that they could. So this is a fluorescence assay where you use this molecule called GHG. That's a marker for the formation of fibers. Amyloid fibers. And you can see whenever you see the signal appearing there, it's indicating that the fibers are forming. And Jill Pike, who was a postdoc in the group at the time, he's back in India now, showed that grass 55, remember the 55? That was different from the our eukaryotes, can also form this fiber. You can see the fiber, this is a TM result of the electron microscopy. We also use flame fluorescence lifetime image to show that there is a signature for the formation of beta sheet rich structures when you do film experiments. And we show that, well, not only that yeast grass can form fibers, but one of the mammalian grass can also form fibers. What for? At that time, I also told Natalia, she was like, well, this is in vitro, right? So how about in the cell? Can GIH1 also form fibers within the yeast? And then she ran this experiment, so you can see on the left, the bright field result is microscopy, right? So that's yeast, those are yeast cells there. And whenever you see a green spot, that's it. And it correlates with the position of these cells. It is showing, all right, that the fibers are forming within the environment, inside the cell, inside the cell, right? When you submit the cell to stress conditions like high temperature or starvation, all right? So whenever the cells are submitted to stress conditions, this kind of fibers form. You can see that happens also in cell, not only in vitro. right? At that time, this was 2019, I came across this paper published, well, this is a kind of editorial piece of paper that this guy had, Jim Rothman, at FFCLRP is one very traditional journal in the area, right? And he has this scientist follow things in Jim's field. Why doesn't this guy has the editorial thing so James? Well, because he's a Nobel Prize laureate, okay? So he won the Nobel Prize in Medicine and Biology in 2012, and here for the studies of the intracellular vascular traffic. And well, this is good to know for this public because Jim is a physicist by training, right? And then he went to biochemist as a PhD student and cell biologist as a postdoc, and ended up winning the Nobel Prize in Medicine and Physiology. So, but he's a physicist. Right? And he was there like saying, look, is the gold stacker phase separated liquid crystal? So he was like challenging the traditional view of the Golgi apparatus I showed you in the beginning of the talk as a membrane bound organelle, well behaved, say, look, that's should be some sort of liquid crystal or colloidal emulsion. And he was proposing that because his group worked with a family of Golgi proteins called Golgins. And Golgins are partners of interactions of grasps. When I saw this thing, I told Natalia in the group, said, look, he's proposing something that the Golgins, these proteins, do in grasps, interact with Golgis. Well, let's see what the grasps can do that too, right? So that's called liquid-liquid phase separation of proteins. I think that many of my colleagues in the field of biophysics of proteins have been sucked into this field called liquid-liquid phase separation of biological condensates. Lisa know about it. I know she told me a bit about it over lunch. So, and one thing that was very obvious to us that the protein can exist in different states, right? So it can be monomeric, soluble there, right? Nothing, and all the way down to amyloid-like fibers. So we saw that grass would be here, and under stress conditions they would be here. But on the way to the amyloid formation, there you can see that there is a liquid droplet state that is possible. So could grasps do that? Could grasp phase separate? If you guys want to learn a little bit more about the history of this liquid-liquid phase separation in biology. So again, liquid-liquid phase separation is a well-known theme in material science, in chemistry, but this is completely new in the cell biology, right? So I point you guys to this piece of paper which was published in Nature in 2018, and it tells everybody how this thing happened, the history of how they found out, the guys who discovered this for the first time found it out. And also point out there because to me, it shows the importance of being in meetings like this, or courses, because the first evidence that the authors of the paper I'll show a little bit later on, found that this phase separation was happening within the cell occurred during a course. They were teaching at the marine biological laboratory there, right? So they started doing some experiments with the students and found out something intriguing was happening with the cell there. And then they went back to their lab and published the first paper on that. Well, the idea of a cytoplasm that was not a pure liquid is old, right? So you can see that E.B. Wilson was proposing this thing. This was a paper published in Science in 1899, and he was there saying, look, the protoplasm, that's the way they call the cytoplasm at that time, was always a liquid, but it's rather a mixture of liquids in the form of a fine emulsion. So he was there saying that, look, the inside of the cell is not a liquid like water in your cup there. So you can see the pictures, right? So it took like over 100 years in the development of my microscopy methods to show, the two guys show the Cliff Brandlin, who is now in Princeton, and Antônio J and Max Planck in Germany, to publish a paper again in Science. This is 2009. A little over a decade ago, to show that this liquid phase separation, the formation of what's called now biocondensates, can indeed happen within the cell. So they show that speed granules are actually a liquid droplets, protein droplets that phase separate from the liquid of the cytoplasm inside the cell. Okay, so if you go through the paper, they show, oh, this is some collection for these instructions. This is a fertilized embryo of C. elegans, and they label the proteins that form as a pea granules with a fluorescent probe. So the green dots you see there, it's the proteins. And when the embryo is at the certain time of evolution, okay, it has to be divided. And so these pea granules, they accumulate in one end of the embryo. So this half of the embryo will be the germinative cells, and the other half will be the constitutive cells of the cell, right? And they show that these green things are actually liquid. You can find several. Oh no, just a second. This is a very cool video they have in the support information of the paper. So that's the nucleus there. Nucleus, and those are the pea granules. You can see that they could just like coalesce in showing that they're like, I mean, there's a whole bunch of data. It's a science paper, so I mean, and the support information is like 3 times the amount of data they have in the actual papers in the support info. So they showed for the first time that these condensates can form within the cell, and it's due to these liquid receptors. After that paper, there was like everybody was looking for condensates within the cell. And from there, like 10 years after that, we can see that a whole bunch of examples appeared showing that condensates can be found in different places in the cell, and they can be formed into the symbol underneath, or they can be there in terms of like a constitutive function, right? So how did we get involved there? As I said, I believe that many of the colleagues in the field were kind of sucked into this phase separation business. Right now, we are not doing that. We somehow found out that the proteins we work with can do that. So I told the guys that in the lab at the time, Felipe was leading this thing, and Rafael and Carol and now doing that. They said, look, how do we induce phase separation in vitro? One thing you have to do is to use some sort of crowding agent. So we went there to the lab and run this microscopy experiment using PEG, PEG, as our crowding agent. If you add a determined amount of PEG, usually 4 to 5% is enough, you can induce phase separation. You can see there, the black kind of a bright field microscopy there. And you can see that the droplets form, condensates form rapidly. And we had to prove that the protein was within the droplet, right? So we labeled the protein with a fluorescent probe and this thing there, and you can see that they can perfectly merge, showing that the protein GRH1 is within that droplet, right? So what does the yeast use this for, right? We show that the droplets can be found, that the condensates can form in a very broad pH range, right? That's important for cell function because the pH changes in the cell, all right? And our idea is that, well, look, remember that I told you guys that RAS is very important for this conventional protein secretion thing, that it goes from the Golgi to back to the ER, and there it somehow engages with a carrier, which transports the molecules to the outside. So nobody knows what carriers are there, right? The best carrier that has been proposed in the literature is this one called CUPS, compartment for unconventional protein secretion. So this comes from the group of Vivek Malhotra in Barcelona, and this paper was published in 2016, saying, look, this CUPS, it's some sort of, it's an array of convoluted network of tubules and vesicles, and they mature. And they are engulfed by this saccule. What is a saccule? I don't know. I don't know what a saccule is. This was the best report. So, right? I was like, oh, my hypothesis is, right now the hypothesis is an in vitro one, because we haven't found a cell biology partner to test this in cell. I think that Vivek will do. I talked to him in January. Well, our results showing that our hypothesis that the cups are actually not a membrane-bound organelle like this one, but it's the condensate formed by blood. So we should form this condensate to recruit the molecules that need to be secreted to the condensate, and then they go to the plasma membrane and then outside. All right. So we upload this results to bioRxiv in December. It was December 19th. I remember the date. So next day, I got an email from Vivek, the guy who said, look, can we talk? I said, wow, gosh, I mean, yes, we are like saying, well, you know, remember the membrane said it was very, I don't believe it is, right? So, but he liked the idea. So he was the review of the paper, which was having just accepted. I know that because he told me, said, oh, I reviewed your paper and I just accepted it as well. Because I think your idea is the best. I was like, yes, right? So, well, but in order, but we don't have it, as I said, we haven't proved that this thing happens within the cell because we don't. Hopefully we'll have a cell biology part soon, bigger. But we could show in vitro that the condensate in vitro can recruit molecules that are secreted to the outside of the cell, right? So, well, GRAS GIH1 can form the condensate. Can it bring a molecule inside the condensate so they can be carried with GRAS? This condensate will be the carrier. So we use this protein called ACPP. So this was the first protein shown to be unconventionally secreted by GIH1, pyruvate mitosis group, same group, right? And we had it in the group and I said, oh, so let's do this the experiment like this. We'll label GIH1 GRAS with red label, and we will label ACPP with a green label, right? We'll test whether ACPP can phase separate, okay, under this condition, and we'll test whether if we form the condensates of a GIH1, whether the two proteins can co-localize within the condensate. Oh, and there you go, great, they can do, right? So if you superimpose the image there, you can see that green and red and green exactly within the concepts of GIH. So GIH1 can indeed recruit ACPP to the condensate, and maybe that's the way this cell uses to secrete things outside. And there is even more interesting thing that brings me to what Lisa has just said, you know, other people has discussed today, which is lots of room here for physicists from material science interested in colloidal motions and this viscoelastic properties of materials, because very little is known about how is the, what are the properties of the inside of the condensate. Can you see that there is a yellow thing there showing, you know, brighter, showing that the inside of the condensate is not homogeneous. What is going on there? Does the structure of the protein change when it's inside the condensate? Nobody knows. Does the viscosity of the inside the condensate change? We believe so. Sure. This condensate can ages. It can transition from a liquid-like state to a solid-like state. That we can prove. But how the viscosity changes with time? So I mean, if you have methods, theoretical or experimental methods, to tackle this kind of problem, this is a very nice moment to be here. There's lots of room for all types of expertise there. All right. Just to sum things up or to finish up, I'm almost done. So well, this was all done with the GRAS protein from yeast. How about the mammalian GRAS? Remember I said there were two. One of them is different from the lower cardiac and the other one. Surprisingly, again, GRAS 55, the same GRAS 55, or same old, can phase separate, can form condensate, and GRAS 65. Yeah. Okay. So this is very interesting because, you know, all the reports of grasps, human grasps involved in conventional protein secretion are related to GRAS 55, not GRAS 65. So it seems that GRAS 65 has a more structural role in the Golgi structure, whereas this one is the one involved in nucleus. And I mean, surprisingly, or curiously, it's exactly this one that can phase separate. This one behind. Right. So we believe we have something there, right? So final slide. I said about carnival like this one, or I mean, we were trying to do some experiments. How about, well, this condensate has to somehow interact with the membrane, right, to be secreted. So let's try to see. Let's see if GRAS 55, the one that phase separates, can, you know, interact with the membrane. So this is a giant level of Pascal, TPPC, labeled with a fluorescent probe. And when we add the GRAS 55, look what it did. I don't know what that is. I mean, I know that those are tubes, right? But I don't know what are the, why? I mean, what? I have no idea. So if you ask me a question about this, I don't say, I don't know. If you have an answer to, you know, try and help me out. right? So we can see that graphite can somehow bend the membrane to produce this. We are trying to look into that, right? So this is it. So but again, the questions there, I think, don't know that there are a few. We don't know the carrier. My guess or my hypothesis that the carrier, UPS carrier, is the concept. Look at there, you can see this is a review paper published in the field years ago. So they say, well, SACU, they put a question mark there, right next to the SACU. So nobody will find. I guess it's the concept, right? Whatever you want to be in this area, I didn't talk anything about applications of this condensate business there, but you can find lots of applications in material science using protein condensate, right? Or if you are in more basic fundamental questions in biology, it's for sure that will be the combination of physics, chemistry, biology, math, computation, math, that type of problem. All right, so with that, I thank you guys for your attention. I also have to thank the ones who did the job, our collaborators in Brazil and outside Brazil, the funding agencies, the group there. So I just want to finish advertising one important event to us. As Danilo said in the beginning, I'm the current president of the Brazilian Biophysical Society. And we'll have this our annual meeting in October here in São Paulo at the Physics Institute at the University of São Paulo. So I invite you all guys to be there, register. There will be 2 symposiums related to phase separation. One of them, both of them organized by guys who are part of this group. So Nelson Oshik will be here. Peter Volnes, whoever likes protein 14. Peter Volnes is one of our panel speakers there. So please visit our website in English. Thank you so much for your attention.
Very good. I stayed here until the end. The subject I introduced with the origin of the genetic code. The 2 first amino acids encoded are glycine and serine that are main components of the liquid phase separation in stress granules. And I brought the data here. We're very glad to know about it. Then I think our papers are the only ones that don't have equations.
Right, I guess so.
And that's where we
I
got
saw that.
the LSPS.
Well, that's one, I think, one of the missions, right? One of the goals to, well, it's kind of still surprising. Those are kind of new results in the field. So, the paper from Brangwin and Hyman came out in 2009. So it's been a little over 10 years, right? So, but it's amazing how many people in the cell biology community still don't know about this thing. So I've been trying to go to cell biology meetings in Brazil. I'll be in the meeting of the Brazilian Cell Biology Society in July. And they just don't know. But, this is a very hot topic there. So there's a guest, Lisa, I think agrees with me, I don't know, that Tony Hyman, maybe Cliff Brayman as well, they are en route for the Nobel Prize. So they are, getting all the prizes that are considered the pre-Nobel awards. So they are getting all of them. And the changes have made it to the textbook. So the Cointanuji book of cell biology, which is like called The Cell, written by Albers, has incorporated in the edition published in 2022, just 2 years ago, has already incorporated these results, considering that the formation of what is called membrane-less organelles is also a way that the cell used to organize the intercellular space. So the community think that they are en route for the Nobel Prize, okay? And I agree, right? So it's the idea here is to look, if you have expertise in things that can help to describe this new, in terms of liquid phase separation, there's nothing new there. The theory, the thermodynamic background that's used to calculate, let's say, the phase diagram of the proteins, you can measure, groups measuring phase diagrams of proteins inside cells. One of them is Thomas Knowles in Cambridge. But the thermodynamic used to describe those phase diagrams is that thermodynamics published by Florian Huggins in the early 40s. It's the same model. And you all agree with me that a protein is polymer is much more complicated than a homopolymer. To start with, a protein is not a homopolymer. And the condensates in the cell have been shown to involve not only one type of protein, different types of proteins, and also RNA. So the results I showed you are pretty well-behaved condensates because just one type of protein, right? So if you think you have the background to tackle this type of thing, this is a very good moment to be in this field, I guess. And then, as I said, there's room for everybody. Everyone. Go ahead.
So you showed that GRASP 55 forms condensates.
Yes.
GRASP 65 doesn't.
Yes.
The first type is composed of many different proteins, right? In the sense that they do not have the same 3D shape. The proteins, you mentioned at some point that
They have very similar 3D shapes.
the first one has similar.
That's the amazing thing, yeah. So that shows us that how dynamics is important to determine the function of a protein, right? It's not only the 3D structure. So we don't want proteins that look like bricks. We don't want proteins like this, right? We want flexible proteins. So in dynamics, flexibility makes a whole bunch of difference. So
But the fact that they have different shapes can be considered a sort of polydispersity of
Yeah.
your collection of particles.
yes.
Yeah, I mean, there are some results that show that if you change only the polydispersity degree of the collection of particles, you can enter in or out the phase separation existence region. So this could be one explanation for
It
Fine.
could be, well, the basic explanation for the phase separation is the one provided by Florian Huggins, which is an entropic enthalpic competition there. So if it's only for entropy, it will be always mixed. But if there are favorable interaction there, so entropy wins, you have the scientific and the mixing each does.
But one of the conditions is low complexity. We
That's right.
that make intrinsically disordered brilliant. Those are the main components of the.
So low complexity domains are sequences of amino acids where you can find low diversity of chemical properties. And that's found in intrinsically disordered proteins. So we believe that RAS can phase separate because they are actually IDR, IgP, right? So that's why. So you need multivalence to provide favorable interaction so that it can overcome the entropy barrier for mixing.
But I may have missed your explanation and a simple question. The intrinsically disordered protein, and then you said it may unfold. So the intrinsically disordered, does it have several specific geometric?
Yeah, well, I always think that there's a problem of the name there. So because when you say disorder, immediately anyone who works with polymers will think that's just a random chain. That's it. But that's not the case when we say intrinsic disorder. That's why I always try to emphasize the intrinsic. So the intrinsic there means that the protein cannot coexist in different organized structures depending on the temperature. And then you can unfold that. And actually, that's one of the signatures we find to determine whether a protein is intrinsically disordered or not, which is force all the conformations to unfold. And then you can say, well, this is unfolded.
No more questions. Thank you again.
Thank you so much.
Thank you all for coming again. We still have a poster section.
Estruturas é muito grande.
Thank you.
Pera, só um segundo, deixa eu só sair aqui. Pera aí, eu não sei. Eu acho que pode ser bastante elevado.
É 1 hora no sábado de manhã, boa tarde, mas em geral a gente gosta de manhã, perto das 10h30, às 11 horas. A média que a gente tinha de 60 a 80 pessoas.
60, 80 pessoas, 1 hora e mais.
Isso. Isso daí, a gente montava os stands de show na física junto, certo? Falou. Até mais.